It's a great pleasure today to receive Professor Maurizio de Pitta. He's a principal investigator of the neuron-glial interaction at the Research Institute of Toronto, Canada. He's an external scientific member at the Basque Center for Applied Mathematics in Bilbao, Spain. Maurizio, welcome.
Thank you very much, Bruno.
So you said you have a lot of new research that has not been published about the glymphatic system. So what are you working on?
Yes, so we are starting some projects in that direction. So let me give you what is the glymphatic system, maybe if somebody want to know. So it's this idea, right? So because in the brain we do not have a lymphatic system itself as we do have peripherally. Now there is this compelling question of how the brain essentially drain away wastes and toxic products of the cell metabolism. The idea is that, again, you have all this kind of massive blood network of vessels, network of blood vessels inside, right? Incredibly elaborated. And at the same time, these blood vessels, right, irrigate and provide nutrients to this mass of brain tissue that brings together neurons and astrocytes, macroglia and oligodendrocytes, and so on. Obviously, these cells, when you look at the cellular level, are like separated, right? They do not really they are not attached. They are like separated by a little bit of protein or like mud or water. This is the extracellular environment. And inside this extracellular environment, there is some fluid, like this interstitial fluid, right? Interstitium, it's in between, right? The cells. Some of this fluid, most of the components of this fluid is water. And so an essential component is water, along with big macromolecules, polysaccharides and lipids, sugars, and so on. And some of this fluid, of course, comes also and is supplied by the cerebrospinal fluid. Now, the original idea was that the cerebrospinal fluid, right, constantly flows inside with this system of vessels and cleans up this interstitial fluid. But it comes at odds because when you have very small space like this, you have also lots of resistance to circulate the fluid. It's like you can think about it when you have a syringe, for example, right? And you put the fluid inside the syringe, right? It takes a lot of pressure in order to put out very quickly all the liquid. Now, inside the brain, you cannot really put a lots of pressure because, I mean, it's a delicate organ, and also cannot be inflated as you want because it's constrained inside your skull. So you need to be very careful with the pressure. The question came up, how do we actually reliably clean or constantly wash out this environment? And so somebody 10 years ago, somebody named Mike Nedergaard, who is one of the actually she conceptualized this idea in a very famous 2-page science article saying the garbage truck of the brain, right? So by simple considerations, she said, so if we look at this environment, right, where the interstitial fluid is, we know that there is a convective motion naturally instaurated by the fact that you have an influx from the arterial part and an efflux from the arteries and an influx from the veins, right? So naturally, this local imbalance between what comes out and what is sucked in, by just the blood pulsation and the heart, creates a sort of a pace, pressure pace, right? That somehow squeeze. You can think about a motion of a jellyfish, right? You can just squeeze. You can think about this motion that pushes away slowly and move the waste. That sounds reasonable, but then what creates and what allows this motion is the water, right? So the cerebrospinal fluid kicks in with this water, but it's not enough. Locally, you need stuff that allows to circulate this water. And what makes this water circulate is a network of molecular channels that are called the aquaporins, that are massive, like massive molecules that literally create holes in the cellular membranes, and they allow this water to, you know, balance in and balance out passively depending on the osmosis and the osmotic pressure locally.
Aquaporin
It's
4, right?
Exactly. It's like exactly a brain fog. Now, what is relevant here is that only one cell expresses these aquaporins, these water channels. And guess what is this cell? It is the astrocyte. So the astrocyte expresses uniquely these water channels. And so things start coming in place, like mosaics, right? They created this kind of extensive sponges, right, where water can go in and out locally and, you know, take advantage of compartments created by their elaborated morphology. And then there is a missing part here that we should take into account, is that the astrocytes also, they literally sit on the blood vessels. They have a part of their shape, of their anatomy, that is specialized, and they literally tile up on the blood vessel. It's actually so essential that it's an integral component of the blood-brain barrier, which brings up the notion of the so-called neuron glial vascular unit, right? Perhaps this construct is one of these efforts we will know probably in 10 years, 15 years, 20 years, 100 years from now. Still, the idea is that we can recognize, right, a modular or like functional unit, which is this combination between the end foot of the astrocyte, or actually many end feet, because an astrocyte can project and have many end feet on either the same blood vessel or very likely multiple blood vessels. So you have an interaction actually through different blood vessels, which are part of a network through the astrocytes. And at the same time, you have an astrocyte, the same astrocyte that can, you know, wrap around multiple synapses, which can be part either of the same circuit or of different circuits. So here it is, you have this kind of middleware, right, the astrocyte, that branches different circuits with the local supply of the blood. And so, even more interesting, it's not only branching because signals, sensing the chemicals, maybe relaying water through the blood inside the interstitial space, and combine with all this idea of the glymphatic thing, right? In which a gly, it's a portmanteau of glia and lymphatic system, right? So glymphatic is the glia through this network of sponges, because astrocytes, despite their name, I mean, they look like stars if you just stain them with the way they used to stain back in the days. But if you go to the ultra resolution, right, the actual cell level or molecule level, which is 1 billionth of meter down, now these folks, they literally look like sponges, like nebulas of many very small branched things. So these sponges, like they literally pile up, they fill up the space, and then they communicate with the interstitial space through these networks of pores that allows the flux of water. Besides this chemical way, they are also able to modify the blood tone, because they actually release chemicals that can contract or dilate the blood vessels. So they have literally something that is able to relay the neural activity or vice versa with the blood tone. And so it's fascinating.
Man. So when you say the Aquaporin 4, they take some of that fluid from plasma, from the blood, then they're going to release it in the, so from the artery, they're going to take that fluid, they release it in the cerebral interstitial fluid, and then reabsorb it in the venous side, the venous astrocyte.
Yes, because so the point is that you have aquaporins also on this end foot, right? And so you have the diffusion, or like the percolation of the ingredients that are responsible for the nutrition, so glucose, for example, or like water or electrolytes that are, the ions that are essential for the signal of neurons, which are potassium, sodium, and chloride, and so on. They percolate through this blood-brain barrier, and they go inside this aqueous environment. But the pulsatile environment, the pulsation of the blood flow, right, creates what is called a difference of pressure that naturally pushes, like the jellyfish, these electrolytes deeper inside the interstitial fluid. And over there, now there is a sort of local gradient of concentrations, difference in concentrations that, of course, when you have more in one area, you try to diffuse to the other area, right? These kind of laws together, they require that you're working into water. It's like you throw an ink drop inside the water. You observe right away the drop, diffuse and dilute. This happens if, of course, you don't steer the water, or you don't have something that create a flow. If you have a flow, and this flow comes from of course, the blood pulsation, then naturally this water, this ink, goes towards diluting towards the flow. Now, what dictates the flow, right, is where you have an opening. And these openings are only provided by the pores that are on the astrocytes. And so the flow navigates in the extracellular space, but then it's regulated by the entry points of the astrocyte. The astrocyte take in or put out, right, this excess of water, and eventually they relay this water through the blood vessels.
And then every arteries and every vein, I guess capillaries in the central nervous system is covered by the end foot of the astrocyte. There's no space. That's a blood-brain barrier. There's like 10 nanometer or something like that.
Yeah.
It's just for water molecule or something like this. And there's a little space around the blood vessel. That's the perivascular space. And that's what they used to call Virchow Robin.
Mm-hmm.
So the same thing. So we have a little space between the end foot of an astrocyte and the wall of an arterial vein. Water is absorbed in an astrocyte, released in the interstitial fluid, and reabsorbed because there's a space in an astrocyte around a vein somewhere else. And so where do we see the cerebrospinal fluid connect with that flow now?
If you go a step back again on, the essential component that is water here in this very narrow environment that is the interstitial splay, right? Now you can think about it, right? For example, when you want to swim, right? If you try to swim yourself or like row, for example, on a boat in a very small container, it's very difficult because all the water that you move, right, will create an enormous hydric resistance. So you cannot really go much. And so this is why often when you want to have this kind of small pools, right, that like are not bigger than a sofa, like you do have actually, yeah. Right, or the bathtub. You cannot definitely swim in a bathtub, right? Not only because as soon as you do a stroke, you probably go out in the living room, but also because if you try to have an extensive, quite a wider bathtub, nevertheless, right, you would have a lots of resistance. So the water can't circulate and you'd be like in trouble. So the same happens in this brain. If you don't have opening ways to, to redistribute the water that you move to go in some place somewhere else, you would have an enormous resistance. So now, because you have this kind of spongy environment by the astrocytes and the pores that these astrocytes have to circulate the water, now the hydric resistance is much less. And so you can not only move things easier from one point to the other, but also in general, you can create flows much easier. So the cerebrospinal fluid, right, which is entering the game here, can circulate in a much more efficient way, eventually reaching everything in an efficient way. So in that sense, you can think about, you still have a contribution of cerebrospinal fluid now in a much more effective fashion, ubiquitously, everywhere, rather than thinking of the fact that you don't have a sponge there. And so it's very harsh to push the fluid to every little narrow space inside.
I read the study in monkey, then, for example, the cerebrospinal fluid in a subarachnoid space is going to have a flow going all the way through inside the ventricle.
Yeah.
So that creates another complete flow in the interstitial fluid that's going to combine with the flow of the astrocyte.
Yes,
so some of
absolutely.
The flow, so some of the water from astrocyte going from the astrocyte on the arteries and the astrocyte on the veins, you're going to have some of that flow that also going to escape to the ventricular system. So you're going to have this mixed flow in the brain, but the CSF is very, very pure. So you don't have to have too much protein there, and it stays very pure in the ventricular system. So you don't want to grab too many stuff, the beta amyloid or whatever, and keep it pure probably by the junction of the parenchyma cells. It's amazing to feel this, the blending of those rivers.
Absolutely. Yes, you can, but you also, so it's from periphery to the center, but if you now think about from the center to the periphery, at the same time, right, the need of generating, replace that fluid, right, it's definitely matched easier because of this way of diffusing faster through this kind of sponge.
Amazing. I totally love that. Wow. They discovered that the lymphatic system expands 2-3 times during the night when you sleep.
Yes, that was a very revealing study. Yes, it's true.
And
yes.
Because of that, they say that's one of for the first time we have a good scientific reason why we sleep. Now we can just open the pathway and drain all the waste product. Before they assume why we sleep, they didn't have a good scientific explanation. That's one of the reasons
I really enjoyed that study a few years ago. Beautiful. One of the first proofs about this sleep regulation of this sponge. Yes, it's.
And then I guess if you are under anesthesia, probably the same thing happens.
Also in anesthesia, yes, absolutely, yes.
So what are you working on with the glymphatic? What are you working with this whole?
So the project that we are running in specific here is understanding how this glymphatic flux is on one hand coupled with the blood, and how the capacity of this glymphatic system could be affected by the electrolytes. Everything that is carried out in our brain is made of charges, right? So molecules and ions do have charges either directly or indirectly because of the electrical fields. And so these charges affect the way things are transmitted throughout the different compartments. And so on the other direction of this project is trying to understand how the actual electrical world that is inside our fluid environment, inside the brain tissue, affects this kind of flow. Electricity still remain something that we are very good at working with. And the only way right now that we have to non-invasively mitigate or modify our brain function, electricity, and together, the coupled system, the magnetic part. So you have electrostimulation or transcranial magnetic stimulation, right? So understanding how this flow is, modifying the general circuits behavior along with the blood also gives you an entry point to understand how to change the magnetic stimulation or the electrical stimulation protocols that we have right now to, modify or improve behavior. This could just be at the level of transcutaneous, right, stimulations, or at the level of more invasive techniques, such as, for example, the deep brain stimulation, for which, of course, surprising technique, right? Works apparently with everything, quite interesting. We don't know why it's working. And it's been on for now 20 years at least. Amazing things. You can deep brain stimulate for now amnesia, deep depression, bipolar disorders, neurological deficits, even dementia, right? With potential for at least rescuing partially cognitive deficits. And still, the reason why this happens are actually not completely understood. And that's because along with the laws of what we are changing and touching there, right? There is also this other fluid component that we tends to completely take out of the game. And now instead, we are recognizing it's very important.
The movement of the glymphatic system and the cerebrospinal fluid are all related to the pulsation, coupled with the pulsation of the arteries. So there is something between the glymphatic system and the lymphatics and the cerebrospinal fluid that now they move more or less at the same time, they connect in the same environment. When you separate, there's something interesting to see there, because one of the rhythm is the rhythm of the artery. What do we have that shows us that the glymphatic system is coupled with the pulsation of the arteries?
Well, there are simple so far. I mean, it's not fully resolved, because if the idea of the lymphatic system would be fully proved, right? Right now, we would have that in the textbooks. Instead, it's still work in process. But you do have what is called correlation analysis, right? So generally, you have a basic evidence for the fact that these opening during sleep or during specific timing of the flow of fluids in the brain interstitial space is somehow correlated or developed in a way that is similar or matches some behavior of the blood rhythm or the cardiac rhythm. Okay, you do have some kind of evidence for that. And as well as locally, you do have correlative evidence for the fact that intracranially, difference in pulsation or in pressure, local pressure, they can actually create this kind of flux. And then you have, of course, mathematical models that suggest and substantiate these claims. Not many, but part of the things that, for example, in my lab we're doing are in this direction.
What are the opposition to the glymphatic system right now? There's some data that go against the presence of a glymphatic system.
More than data is technology that it's hampering, because I am a strong supporter of this idea, of course, and I think it's fascinating and it does definitely make a lot of sense. I would say that mostly we do not have enough technology here to harness a specific astroglia or glial functions, right? So we only rely on ways, right, that are proxies of these activities so far. When it comes to humans, it's only mostly on a speculative point. And the reason is because the same technology, right, that we use right now to image these responses, so which is these optical tools, right, that they look and the fluorescent on the chemical signals. And unfortunately, those optical tools, they are not applicable in humans because they kill the tissue on the short run. Functional magnetic resonance or PET scanning is something you can do every now and then, right? Because it's not great to give your patient every day a dose of radioactive substances that enters in the brain and then just irradiate for a while.
What about diffusion tension imaging? Is hard to work with? Because that's specifically
No. Diffusion tension imaging has been used, and I mean, it's also very nice. DTI. They don't give you this kind of resolution, which in the brain means what is the source of these alteration. They don't give you this kind of resolution, which in the brain means what is the source of these alteration, except in specific cases. I mean, you can, for example, definitely guess informations on white matter alterations, right? But at the level of brain activity and the astrocytic component of this brain activity, no way to get so far a non-invasive technique that can actually give you an information about the activity of these cells.
Maurizio, it's fascinating. I'm so happy with this discussion. And
thank you.
Yes, and I hope you have a great success with your continuing study, and I'll be happy to read you more and more in the future.